Lesson 5 has to do with sex-linked traits. So these are traits that are specific to the X and Y chromosomes that are passed on by the mother and father. Okay, so a sex-linked trait is a trait that's coded for on the sex chromosome, the X or Y chromosome. So when we looked at all those homologous pairs of chromosomes in previous units, there was always at the very end an X and Y or X, depending upon whether it's male or female, chromosomes at the very bottom right hand side of those homologous traits. So if a gene's located on the X chromosome, it's called X-linked. If it's on the Y chromosome, it's Y-linked. The X chromosome is larger, therefore there are more X-linked than Y-linked traits. An example of an X-linked trait is red-green color blindness in humans. If you can't make out the number that's on here, then chances are you have some issues with the red-green color blindness or red-green color identification. There's other types of color blindness too, and this type of format allows people to determine whether somebody's colorblind or not. Very easy way for them to tell with young kids as to whether they can see the two different colors on there. Because for somebody who's colorblind, they would not be able to see an image in this. Okay, so Punnett squares. When you're doing sex-linked Punnett squares, the X and Y chromosome need to be included to show which chromosome they're on. So, we're going to have to keep track of the sex of the offspring and the trait that is linked with it. So notice in this one, the male does not contain the trait. So the Y chromosome is specific to males. Female is XX, male is XY. So the males can get the X-linked traits, and they can get them a lot easier. So males only need one recessive allele for the trait to be expressed, and that's why color blindness, with it being sex-linked, is much more common in males than females. So if we look at this, if we have a normal female and a colorblind male, the capital C here represents the color blindness. And here's another the capital C is the dominant, which means there isn't color blindness. And then the lowercase c represents the color blindness because we have the colorblind male here. So we would end up with a big little for the first female and a big little for the second female. We would end up with a big for the first male and a big for the second male. So in this case, every single one of the offspring would be normal, even though the dad's colorblind. Now, if the mom carries the trait, it gets more shady. That's where we can get the offspring that have the color blindness. So let's look at a male who has the color blindness with a female who is normal but is a carrier. It takes a lot for females to be colorblind. So if we go our X, and she is a carrier, so we're going to have a big C, and we're going to have a little c. And then on our male, we're going to have a little c and our y. So if you go ahead and cross these, you're going to have the big C from the mom up above. We're going to have the little c from the dad. We'll have the little c from the dad. And we'll have the little c from the mom. So this one's going to be colorblind. We will have the big C from the mom. We'll have the Y from the dad for the first boy, so this one will not be colorblind. Then for this one, we'll have the little c from the mom, the Y from the dad. So since this one only has one X chromosome, this one's going to end up being colorblind. So based on this cross, because the mom's a carrier and the dad has color blindness, this is actually the only way you're going to get a colorblind female. The mom would have to be a carrier for the trait, and the dad would have to have the color blindness trait. So when we talk about sex-linked genes, we generally ask about, okay, what are the odds of the child having color blindness? And then what are the odds of a boy having the trait or a girl having the trait? So you'll see questions like that. If you have questions on this, let me know. But this is one where you do have to put the X and Y chromosomes just because the fact that the one chromosome is not carrying it. So we need a placeholder so that we know what's going on with the Punnett square.